The formation of the embryo sac or the egg apparatus inside the ovule is what is known as the process of megasporogenesis. It is usually that in that particular ovule, we will find that inside this nucellus region, the nucellus which is made up of parenchymatous cells, one of the hypodermal cell it actually transform into this particular structure. So what actually happens is that the primary archosporial cells, which is nothing but a functional hypodermal cell, it actually starts meiotic division and forms this like primary parietal cell and actually it's written both as a primary parietal cell, but in reality it is like the top portion is a primary parietal cell and the bottom one is called the primary sporogenous cell. These two cells are developed. This is by simple peritoneal division of this particular cell. So what we are going to find here is that this particular structure, the one that is formed here, is what is going to lead to the formation of the megaspore, or we can simply say that this archosporial cell is what is going to be transformed, or what is the one which is actually can be considered as the microspore mother cell at the moment. Now, the parietal cell after the division actually like forms the parietal tissue, the one that surrounds the entire thing. Whereas the sporogenous cell is actually the one which which starts the division of meiosis and undergo like division like two times, as you know, like this is the one which is transformed into like this is the one which has formed into the megaspore mother cell. Now that is the one which is now dividing several times, like in the sense like in the process of meiosis as it happens it divides twice. So by that division, it actually produces the haploid megaspores. All four of them are actually haploid megaspores. The parietal cell is at the base of it, at the right at that particular location. And you can clearly see it is right there that the integument has developed and it has like trying to cover that area. That means this region is actually the micropylar end of the entire ovule. So that means out of those two cells, the cell which is at the chalazal side, means the cell which is towards the chalazal side is the one which behaves as the sporogenous cell and is the one which actually divides further into four haploid megaspores. But this four haploid megaspore, it doesn't remain functional as it is, because out of this four, three of them will actually degenerate, because only one of those three will be behaving as the megaspore, which will later be transformed into the main female gametophyte form names, the main gametic cell, we can say. So for that particular reason, it is this particular one out of the entire lot. Again, the one towards the chalazal side means this one, not the other three. Is right. This one is the one which actually develop, or the one which actually transform fully into the functional megaspore. The rest three get degenerate. As you can see, it is being shown here that the rest three has got completely degenerated. So what we are left with is this functional megaspore. Now this megaspore will actually undergo mitotic division to lead to the formation of the final structure, which is the egg apparatus, which is this one. So for that, it will actually divide three times: division 1, division 2, and division 3. Three times it is actually dividing. All three times it is mitotic division, because already it has been transformed into a haploid cell or haploid nucleus right here in that region. Now, in the mega gametogenesis step, it is actually like this cell is now undergoing simple mitotic division, because it cannot further reduce the chromosome number into even more half, so it is not possible. So it is dividing mitotic way, And now we find that it is making 2 copies, 4 copies, and finally 8 copies of the haploid nucleus that it actually got. Now, finally, what will happen is simple rearrangement. Rearrangement of all those cells, or sorry, all those nucleuses to a certain side, resulting in formation of this particular final egg apparatus, where the three nucleus like will be moving towards a pair of three nucleus will be moving towards either like polar side. So it's like 3 here and 3 there. The remaining 2 will be localized at the center. So this one at the center will actually be the polar nuclei, which will fuse together to form the primary endospermic nuclei later on. And this other 3 that are there, sorry, the other 3 that are there, so the other 3 actually, the one towards the micropilar end will behave as the main egg apparatus, the one responsible for the production, or the one we actually will have the egg cell with the synergids, where its exact polar side means the antipodal cells, the one that are formed, the antipodal cells, are always arranged towards the chalazal end. So that is how the megagametogenesis process takes place. Here is another representation of the entire thing with a simplified diagram, and you can see the final like development of the mature female gametophyte is taking place right there. Now keep in mind the mitosis division that is taking place in the megagametogenesis process. All these three mitotic division, it is not like the process is complete. It is actually only the nuclear division takes place, not the cellular division, because the final cellular division takes place when this entire gametophyte is actually now transforming into the mature female gametophyte. At that time the cytokinesis takes place, because that is when the entire cell gets divided into those 7 cell structure. Now, uh, until that point, we will find it is just simple karyokinesis taking place, and it is like just simply separating all the nucleus. Only that much. Now, this particular form of megasporogenesis and megagametogenesis process is actually what is known as the monosporic type of megasporogenesis process, we can say. Now this process, it is like very common. Most of the organism, like nearly 70-75% of the plant like angiosperm species, even many of the gymnosperm species actually follow this particular method. And this is also known as, or goes by the name of polygonum type of sac formation. So where we find this is the normal generalized arrangement. This is the one we actually majorly focus in our within our syllabus, scope of the syllabus. The other two methods that actually exist are the bisporic type and the tetrasporic type, where you can see it's simply like the division is like in this case, we find that the female megaspore is actually one and the remaining 3 degenerate. Here, 2 of the nucleus becomes the female like megaspore, but one it actually like here the separation is like 4 cells are forming here. Here only 2 cells with 2 sets of nuclei, haploid nuclei is there. So one of them actually remains as it is, the other one it degenerates like that. So this final megaspore is now carrying 2 nucleuses, 2 haploid nucleuses there. So when that enter the process of megagametogenesis, the division is slightly different. It is like one, and it is like 2 mitotic division will take place, and now there will be arrangement of only the egg apparatus and like the polar nuclei. There are 3 nuclei that will be found present inside. Um, the other type is actually the tetrasporic type. So in tetrasporic, what we are going to find is that the division is taking place as like the normal meiosis division as it is supposed to happen, but the cytokinesis will not occur. It is only karyokinesis process will occur there, which result in like formation of all total like 4 nucleuses, haploid nucleuses inside that cell, where 3 of them cluster towards one corner and one of them towards the other end. So the one at the other end, the solo one, is the one which transform into the egg apparatus later on by mitotic division, which will again be like only two sets of mitotic division taking place, not three. Finally, resulting in formation of the egg apparatus, as I mentioned at the bottom. One centralized like nuclei at the very central nuclei having like pair of nucleus is there, and at the same time there will be a numerous number of antipodal cells towards the other corner. So that is how the like tetrasporic type of division or tetrasporic type of embryo sac formation actually it happens in the in many of the like species of plants. But the one that is very much common is actually the monosporic type. The very common, the one that we see all around in every major type of plant family that we know about. So finally, this mature embryo sac will be developed, and this mature embryo sac it actually has like the most important part, which will be the egg apparatus, comprised of the egg cell and the two synergids. Keep in mind the synergids are actually responsible for the like partial nourishment and entry, mostly the entry of the pollen tube through this area, for which this filiform apparatus we can see right at the bottom, the filiform apparatus is majorly responsible for it. So what actually happens is when this filiform apparatus arranged towards the micropylar region, so the pole like the like the pollen tube when it comes, it actually enter through this area, and let's say this marker being the nucleus, the sperm nucleus, it actually go and fuse with or fertilize the egg cell, and another one it goes and fertilize the polar nuclei. That way the fertilization actually takes place there. So its major function, majorly like the synergies are responsible for this entry of the pollen tube. So for that, it actually secrete chemicals so that the pollen tube can be directed towards that side. It does provide nourishment to the embryo sac initially, like it does provide the early nourishment to it. And synergies they do get degenerate after the fertilization of the egg takes place. Now, egg, as it is the main gametophyte, you can say the one responsible for the process of formation of the zygote. So it will be the major, the most important nucleus out of the entire lot of all the cells. Then we have the central cell, which is nothing but a very like large structure carrying two polar nuclei inside, and these two polar nuclei, which will fuse with another the sperm nuclei, and ultimately will lead to formation of the primary endosperm right here in this particular region. And this primary endosperm is a nutritive structure which provides nutrition when the embryo is developing, means when the zygote is developed into the embryo, the seed this embryo is about to germinate out. At that time the early nutrients are being gained from this endosperm itself. So it is a very important like thing out of the lot. Finally, the antipodal cells, all three cells at the very corner, these antipodal cells, they are actually uh, like they limit the chalazal expression, means it doesn't allow the fusion at the chalazal end, means the always it is directed towards the micropylar, is going towards that side and entry through that uh, end, finally fertilizing the egg cell. But it is this region which actually prevents the this pollen tube from entering that area. To some extent they are responsible for emitting some sort of growth factor for the developing of the embryo sac. That is the only thing that happens. But after the fertilization, these three cells also they get slowly degenerated and completely destroyed. So ultimately this is the egg apparatus which will post the fertilization, it will transform into the seed, and which will be carrying the embryo inside it.